A pleasant day. I have a situation for you, class. You have a patient that needs a blood for transfusion but doesn't know his blood type. The phlebotomist took sample from the patient and gave the sample to you. After doing the forward and reverse grouping, the reaction of the anti-D was negative. What will you do next? Will you release the result as Rh negative? Well, the answer is no. You will not report it as Rh negative directly. You should test it for weak D antigen or DU first. Why? This will be our lesson for this week. Test for weak D antigen. At the end of this week, students are able to discuss the mechanism of the expression of weak D antigen. Explain the implication of the expression of a weak D status or the DU phenotype. Create a scheme to detect the presence of weak D. Enumerate the equipment and materials needed in the detection of weak D antigen. Most D-positive RBCs react macroscopically with anti-D at immediate spin. This is referred to Rh-positive, which have a 3 plus or 4 plus agglutination reaction. However, there are some D-positive RBCs do not react or agglutinate at immediate spin. That is the reason why we need to test for weak D antigen before releasing the result. There is a need for further testing at 37 degrees Celsius or AHG phase to determine the D status of the patient. This happens when the individual has a weak D phenotype. Weak D phenotype is a weakened form of D antigen. Individuals having this phenotype have a weakened expression of D antigen because of the variations of the quantity of D antigen or in the specificity of D antigen epitopes. Mistyping of Rh may cause hemolysis of red blood cells and may cause death to the patient. We don't want that to happen. Weak D test determines the presence or absence of DU factor or the weak D antigen on red cells. Weak D antigens react strongly in indirect antiglobulin testing phase. Question: when will you process a test for weak D antigen? You will process for DU if the anti-D reaction in the forward grouping is negative or have a 2 plus agglutination. So, if you have a 2 plus or less reaction on anti-D anti-sera, proceed to the weak D test. We will also do this test when there are Rh typing discrepancies between current and previous results. And lastly, we'll do this when neonates are born from Rh negative mothers. If the weak D test of the baby is D positive, the mother will be candidate for RHIG, so that the next baby of the mother will not have hemolytic disease of the fetus or newborn. Another question. Bakit may mga individuals na weak ang expression ng D antigens nila? Answer. Weak D arise from three mechanisms: the position effect, quantitative, and a partial D antigen. Position effect or gene interaction effect happens when C is in trans to D. Meaning the allele carrying RHD is in opposite haplotype to the allele carrying C. For example, in this manner. So, in the chromosome, the position is in trans. The position effect does not happen if the C is in cis position with the D, like this. In cis position, in chromosome, this one. In this case, the D antigen is structurally complete, and these individuals still D positive in immediate spin. Individuals having this mechanism can be safely transfused with D positive blood components. Quantitative changes results from inheritance of the RHD genes that codes for a weakened expression of D antigen. In this mechanism, the D antigens expressed are complete but fewer D antigen sites. Kokonti lang yung D antigen sites kasi may mutation na nangyayari sa RHD gene. These mutations will cause changes or deletions to the amino acids present in the transmembrane or intracellular region of the RHD protein. These protein changes happens inside the RBC. The RBCs agglutinate weakly or not at all in immediate spin phase, but strongly agglutinate in AHG phase. This phenotype rarely produces anti-D since RHD antigens are still present but fewer in number, so we can safely transfuse D positive blood components. Partial D is a mechanism where one or more epitope or part of the D protein is missing or altered. It is also called as D mosaic. In this mechanism, the D antigen is not complete. RBCs of this type weakly or do not react at all in immediate spin. These individuals are type D positive and produce anti-D which agglutinate or reacts in all D positive RBCs, but do not affect their own cells. The anti-D produced by these individuals may cause hemolytic disease of the fetus and newborn and transfusion reactions. Rh negative blood should be transfused to these individuals. So class, this is a schematic representation of antigens and epitopes on weak D partial D, and a normal D positive red cells. So number 1 shows a normal D antigen with all its epitopes. These colored ones are the epitopes, and as a whole, this is the D antigen. So ito, sa number 1, kompleto yung antigenic sites and kompleto din yung epitopes ng D antigen. Number 2 is weak D with reduced D antigenic sites and all epitopes are present. So, sa napapansin nyo, tatlo lang yung antigenic sites. And then, kompleto pa rin yung epitopes. Number 3 is the partial D with normal D antigenic sites but lacking 1 D epitope. So, normal lang yung antigenic sites pero kulang ng isang D epitope, yung D antigen. Next is the partial D with normal D antigenic sites pero may kulang ng dalawang D epitope. And lastly is the partial weak D with reduced D antigenic sites and kulang ng isang D epitope. So these are the RHD variants. So if the forward typing with anti-D is negative or no agglutination at immediate spin, incubate the tube at 37 degrees Celsius for 15 minutes, but in some laboratories for 30 minutes, and then centrifuge for 15 seconds, and then observe for agglutination. Presence of agglutination is positive. So, if there is agglutination, report it as RH positive. If still no agglutination, wash the tube 3 times with normal saline, add 1 drop of AHG, centrifuge it for 15 seconds, and then observe for agglutinations. Presence of agglutination is positive, so meaning there is a weak presence of D antigen. So, if there is still no agglutination, add 1 drop of Coombs check cells. RBCs, check cells are RBCs that are coated with IgG. These cells are used to check if you perform the weak D test properly. Then, observe for agglutination. If there is presence of agglutination, it indicates that the test is properly performed. No agglutination indicates that the test is not properly performed, and the test should be repeated. If properly performed and still negative, then the result is RH negative. Note, patients or recipients that require AHG testing to determine the presence of D antigen and have the D antigen are the weak D positive. This is the guide in the interpretation of tests for D antigen. So, anti-D in slide or tube method, if this shows a positive or with agglutination, the manner of reporting is RH positive. So, meaning positive reaction indicates the presence of the D antigen. If there are no agglutination, so it is initially Rh negative. So ang gagawin, negative reaction should be further tested for the presence of the weak D or the use of AHG, with the use of AHG. So, AHG test for weak D or indirect anti-human globulin test. So, if the DU reactions is 0 or no agglutination, so you will report it as DU negative. RH neg or DU negative. Negative reaction confirms that the person is Rh negative, patient or donor. But if the DU reaction is positive or with agglutination, so the manner of reporting is DU positive. So Rh neg, DU positive should be given proper classification. So if patient siya, ang result niya is Rh negative. If donor, Rh positive. Class, take note that if the reaction is positive for DU test, the person or the patient should be given proper designation. If this individual is accidentally given a Rh positive with complete D antigen blood component, he may develop an antibody and this should be avoided. Always remember that DU positive individuals are classified as Rh positive when they donate blood and Rh negative when they receive blood. The goals for week D testing are to improve accuracy in Rh typing, conserve Rh negative units which will be given to those who need them, and prevent unnecessary administration of Rh immunoglobulin in weak D stereotypes. Exercise 12, testing for weak D antigen. At the end of the exercise, you are able to perform a test for weak D antigen with great accuracy. Recognize a significant agglutination from a non-agglutination reaction. Grade the agglutination reaction. Determine the presence or absence of the weak D antigen. And determine the blood type as Rh negative, Rh positive, or DO positive. The materials or equipment needed for this exercise are Wassermann tubes, marking pen, centrifuge, pasture pipettes, test tube rack, gum label, gloves, and Nasco film. The reagents or samples needed are anticoagulated blood samples, anti-D, anti-human globulin sera, normal saline solution in a wash bottle, and 22% bovine serum albumin. For the procedure, number 1, prepare 2% to 5% red cell suspensions to be tested. Number 2, label another set of tubes as U and NC, U for unknown, NC for negative control. Number 3, follow the table below. So, anti-D, add 1 drop in unknown, to 22% bovine serum albumin, add 1 drop in the negative control, and 2 to 5% red cell suspension, add 1 drop on each tube, unknown and the negative control tube. Number 4, mix gently and cover all tubes with Nesco film. Incubate both tubes in 15 minutes at 37 degrees Celsius water bath. A known tube with agglutination is regarded as Rh positive. A known tube without agglutination goes to the next procedure. Number 5, wash the cells 3 times with normal saline solution. Number 6, decant the saline completely after the final washing. Number 7, add 2 drops of anti-human globulin reagent to all tubes and mix gently. Cover with Nesco film. Number 8, centrifuge for 15 seconds at 3,400 rpm. Number 9, gently dislodge each cell button and examine for agglutination. Number 10, interpret and record the results. Note, the absence of agglutination confirms the blood group to be Rh negative. The presence of agglutination means the presence of weakly reacting D. Number 11, for each negative tube, add 1 drop of well-mixed Coombs check cells. For the laboratory output, watch the video links and answer the lab activity. A PDF will be provided for your lab activity. For the observation part of the activity, base your answer in the videos. Submit your activity in PDF form. The format of the file name is section, last name, and exercise 12, or as preferred by your professor. Digital drawing is allowed. Thank you and have a great day.